Some of the most dangerous bacteria in the world are incredible masters of disguise. They hide behind this wall that's almost impossible to get through. But for over 100 years, we've had a really clever way to unmask them. We're talking about the Ziehl-Neelsen stain. It's a foundational lab technique that literally makes the invisible visible, and it's a huge weapon in the fight against diseases like tuberculosis. All right, so here's our game plan for this explainer. We're going to start with the science behind that waxy fortress, then dive into preparing for the stain, the staining protocol itself, how to read the results, and, of course, making sure it's all accurate. We'll wrap up with a look at the bigger picture and some alternatives. Let's jump in. Okay, first things first, we gotta understand our opponent here. The whole challenge with mycobacteria comes down to their cell wall. It's unique, and it's incredibly tough. Seriously, this is not your run-of-the-mill bacterium. Think of it more like a tiny biological tank. So that leads us to the fundamental problem, right? So the very question this technique was invented to answer. If you've got a bacterium that shrugs off most of the stains you throw at it, how in the world do you make it visible? How do you stain something that seems, well, unstainable? And the secret ingredient to all this resilience? It's a substance called mycolic acid. I mean, just look at it here. It's this waxy, fatty armor plating. This layer makes the whole cell wall hydrophobic, which is just a fancy way of saying it hates water. So when you try to use a normal water-based stain, it just beads right off. That's why it's so hard to see. And this is where the genius of the Ziehl-Neelsen method really shines. It's this elegant three-step attack. First, you hit it with a primary stain. You use heat to literally force a powerful red dye, carbol fuchsin, right through that waxy wall. Then you decolorize. An acid alcohol wash comes in and strips that dye from everything else. But the mycobacteria? Their waxy armor just won't let it go. And finally, the counterstain. You add a different color to stain all the background stuff, making our bright red target just pop. Okay, so we've got the theory down. Now let's get practical and head into the lab. And you've got to understand, a great stain doesn't start with the dye. It starts way before that, with super careful preparation and a massive focus on safety. And when I say safety, I'm not kidding, especially if you're handling something like sputum, which could be infectious. These rules are absolutely non-negotiable. You're working inside a biosafety cabinet, you've got your gloves, lab coat, eye protection, the whole deal. And fixing the smears is crucial to cut down the risk of creating aerosols you definitely don't want to be breathing in. Getting an accurate result really comes down to creating the perfect smear. It's kind of an art form. You start with a pristine, grease-free slide that's labeled, of course. Then you carefully spread your specimen into a thin, even layer. You don't want it too thick or too thin. Let it air dry all the way. Then, for the final touch, you do what's called heat fixing. Just a couple quick passes through a flame. This glues the specimen to the slide and adds another layer of safety. Okay. Safety checks are done, our smear's prepped and ready. Now it's time for the main event, the staining protocol itself. And let me tell you, this is where precision and timing are absolutely everything. So, here we go. First, you flood the whole slide with that red primary stain, carbol fuchsin. Then comes the most important part. You heat it, gently, just until you see steam rising. You keep that going for about 3 to 5 minutes. That's what drives the dye into the cell. Then a good rinse with water. Next up, you decolorize with acid alcohol, and you keep going until the liquid running off is totally clear. Rinse it again. And finally, you apply the counterstain for just 30 to 60 seconds, do one last rinse, and let it air dry. This table really breaks it down nicely, doesn't it? It's like the recipe card for the whole process. You've got your 3 to 5 minutes of heating with the carbofluorescent, then the decolorizing step, which is usually 1 to 3 minutes, but the real key is to watch until it runs clear. And then the counterstain is super quick, just half a minute to a minute. See? Every single second really does count here. All right, all the wet work is done. The slide is stained and dry. Now for the best part, the moment of truth. We're heading to the microscope to see what we've got. This is where we interpret the results and turn a colorful slide into a really important report. So what are we looking for exactly? Well, under the microscope, usually with an oil immersion lens, it's all about the color contrast. Our targets, the acid-fast bacilli, or AFB, are the ones that held onto that first dye. They'll be these bright, almost glowing red rods. Everything else, all the other cells, the debris, that all got decolorized and then picked up the second stain, so it'll just be a blue or green background. But it's not just about finding them, it's about counting them. Just saying it's positive isn't enough for a doctor. they need to know how positive. So labs use a standardized scale, like this one, to grade the result. It gives a sense of the bacterial load, right? Are we talking just a few bacteria here and there, which is scanty, or is the slide loaded with them, what we'd call a 3+ result? It's crucial information. Okay, this next part is huge, because none of what we've talked about means a thing if the result isn't reliable. So let's talk about quality control and the common pitfalls, because getting this right is just, well, it's mission critical. Think of this like a pre-flight checklist for every batch of stains you run. You absolutely have to validate your work. That means you run a slide you know is positive and one you know is negative right alongside your patient samples. You're also checking the basics. Are my reagents expired? Are the slides totally clean? Did I heat it properly? Did I decolorize for the right amount of time? You have to take every single box. And the consequences of getting it wrong? They're huge. Look at this. If you decolorize for too long, you wash the stain right out of the target bacteria. That's a false negative. You tell someone they're clear when they're not. Don't decolorize long enough? Now you've got a false positive, potentially starting someone on heavy treatment they don't need. Make the smear too thick and you can't see anything clearly. Not enough heat and the stain never even got in. Every one of these mistakes has a direct and pretty dramatic impact on a real person's diagnosis. So the Ziehl-Neelsen stain is a powerhouse for sure, but it's not the only tool in the toolbox. Let's zoom out a bit and look at the bigger picture to see where it fits in. Okay, so you've got the classic Ziehl-Neelsen, the hot method we've been talking about, which uses heat. Then there's an alternative called the Kinyon method. It's a cold stain, meaning no flame. It gets around the heat by just using a way more concentrated stain. And then you have a completely different approach, fluorochrome staining. This one uses fluorescent dyes, so the bacteria literally glow in the dark under a special microscope. It's way faster for screening a ton of samples. So what's the bottom line here? Where does this stain fit into a real-world diagnosis? Well, think of it as a crucial first piece of the puzzle. It's a fantastic screening tool. It's fast, it's cheap, and it gives you a really good initial idea of what's going on. But it's not the final word. A definitive diagnosis always means putting that stain result together with the patient's symptoms and then confirming it with a more specific test, like a culture or a molecular test. In the end, it all boils down to this, doesn't it? This isn't just some abstract lab procedure. Mastering this technique, really getting it right every single time, that directly translates into finding the disease earlier, reporting it more accurately, and at the end of the day, giving patients better care. It's as simple as that. I mean, the Ziehl-Neelsen stain is all about figuring out how to see something that does not want to be seen. It makes you wonder, right? What other invisible worlds, what other microscopic secrets are just out there, waiting for someone to invent the perfect key to unlock them and bring them into the light?